Good morning, good afternoon, and good evening to you all. On behalf of the Secretariat to the Convention on Biological Diversity, I'm very pleased to welcome you all to this webinar to launch our latest technical series on synthetic biology. To help us mark this achievement, we have arranged this virtual webinar where the authors of the updated technical series will introduce the updates and share the latest information on synthetic biology. We are also very privileged this morning to be joined by the Director of the Science, Society and Sustainable Futures Division in the Secretariat, Ms. Ji-Hoon Lee. She's joining us today in support of this work and on behalf of the Executive Secretary, and will deliver the welcoming remarks. So without wasting any time, please help me to welcome Ms. Lee. Ji-Hoon, over to you.
Thank you, Wachi. Can you hear me?
I can hear you, we just can't see you.
Oh, I can see myself.
Okay, perfect.
Okay,
Over to
good.
you. Thank
Sure,
you.
Good day to all of you. Welcome to this very important event being convened by the CBD Secretariat. Today, we celebrate with you the launching of the new publication, the Technical Series number 100 on synthetic biology. This publication has been prepared by the Secretariat in response to a request from the Conference of the Parties in its decision 1419 to update a previous publication on the same topic released in 2015 as CBD Technical Series number 82. For this purpose, the Secretariat has partnered with the International Center for Genetic Engineering and Biotechnology, and I wish to express our sincere thanks to our partner for their valuable contributions. Ladies and gentlemen, synthetic biology is a rapidly evolving field that has generated interest and also concerns among numerous industry sectors, academia, government agencies, intergovernmental organizations, and civil society groups, including parties to CBD. CBD parties have demonstrated their interest in the subject of synthetic biology with discussions going on for over 10 years, especially when it comes to potential impacts on the three objectives of the convention. Moreover, the Conference of the Parties has emphasized the need for coordinated and non-duplicative approaches on issues related to synthetic biology under the convention and its protocols, as well as among other conventions and initiatives. Dear participants, synthetic biology has a potential to offer a cure for cancer, solution for epidemics, reduce carbon emissions, and address many other pressing environmental and biodiversity-related challenges. For example, during global pandemics, we have learned that horseshoe crabs have been integral to the safe production of vaccines. This process, however, raises a lot of concern on biodiversity loss and ecological sustainability. That's why we note with great interest some of scientific studies showing that synthetic alternative can substitute for horseshoe crab blood. However, as is the case for any technology, the use of synthetic biology may also come with risk, which, if not carefully addressed, may lead to adverse impacts on biodiversity, health, and the environment. Ongoing global debate regarding synthetic biology, therefore, covers a broad range of social, ethical, and economic considerations regarding synthetic biology application and tools. Ladies and gentlemen, this new publication, Technical Series number 100, will update you as an information one-stop shop regarding supporting technology for synthetic biology, the state of development of synthetic biology applications, potential impacts of this application on biodiversity, socio-cultural consideration, and the complex international debate on regulatory and governance issues. This publication comes at the high time, given the ongoing negotiation process under CBD on the development of post-2020 Global Biodiversity Framework, or GBF. In the discussion on GBF by parties, synthetic biology and its associated technologies are viewed as part of a larger collection of the tools and solutions to reduce threat and meet people's need. Promoting and ensuring their safe use will help us achieve the 2050 vision of living in harmony with nature by offering opportunities to take advantage of this technology towards conservation, restoration, and sustaining a healthy planet. Distinguished participants, with great pleasure, the Secretariat presents this updated report in Technical Series number 100 to all of you, together with a representative from International Center for Genetic Engineering and Biotechnology and other experts from parties and partner organizations. We hope you greatly enjoy it. Thank you.
Thank you very much, Jihun, and thank you for opening the webinar and for those welcoming remarks that just emphasize how excited the Secretariat is to launch this publication and how we see this publication contributing to the body of work on synthetic biology and the state of the art. So thank you very much for joining us and for those remarks. We will now move on to the technical parts of this launch, but before I do so, just some housekeeping. Kindly note that this webinar is being recorded and will be made available on the Biosafety Clearinghouse. If you encounter any technical problems during the course of the webinar, please contact the Secretariat and we will attempt to assist you. There's a question function in the control panel. Please feel free at any time to raise any questions, comments, compliments in the questions panel. And at the end of the session, when we've heard from all the presenters and the respondents, we will then go through the questions and attempt to address them. If you want to refer to a specific presentation for your questions, please do so as well. This technical series has been a phenomenal project, as Ms. Lee has indicated, and we are certainly very proud to present it to you today. And we will now start with the first presentation by Ms. Marianela Ara, the Program Officer responsible for synthetic biology in the Secretariat, and she'll provide us with an introduction and background. So Marianela, over to you.
Thank you very much, Watsi. Let me just ensure. Can you see my presentation now?
Yes, your presentation is clear.
Right. Thank you very much, Watsi, once again, and welcome everyone to the launch of this technical series number 100. The first presentation today, it's meant to share with you some background information about the update, as well as some information on the process that we follow for doing this update of the technical series. And then after this presentation, you will be also hearing from the authors on the content of the various sections of the document. So let me start with some background information. As was mentioned by Jihun previously, this technical series number 100 is an update of an existing publication that we had in 2015. That was technical series number 82 on synthetic biology. And during COP14, through decision 1419, the Conference of the Parties requested the Secretariat to do an update of that technical series. So with financial support from the governments of Austria and the Netherlands, we started the process of updating the technical series in November 2020. And we did that in collaboration with the International Center for Genetic Engineering and Biotechnology. So in terms of the process, the update was prepared mainly by the 3 lead authors from the ICGB, and inputs and guidance were also provided from the contributing authors from the Secretariat. So it was a process where we worked very closely together. The update took us over a year. As I said, we started in November 2020, and we are launching the document now. And we basically work on this update through various phases. The first one was pretty much like a brainstorming session where the authors and the Secretariat staff started discussing how we wanted to do the update, what kind of parts or areas of the previous document were needed to be kept, basically which areas were in need of an update or should be replaced because they were no longer valid or things like that. So once we went through that thinking process, we came up with an idea and a plan to do the first draft. We embarked on the process of doing the first draft, and that first draft was presented to the SAPSTA meeting. That was the SAPSTA 24, the first part of the meeting, which is the one that was virtual, and it was presented as an information document to that meeting. Almost at the same time, we opened a peer review process where we call for comments on the first draft of the document, and I will comment on the peer review briefly soon. And then after the peer review, of course, then we consider the comments that we received, and we embarked again in the process of drafting the final version of the document, which has been now edited, and it's the one that is available on our website. So for the peer review, I want to highlight that for us, it was a very, very useful process. We received a substantive amount of comments from various sectors and actors, and I think the information that we received was very valuable for us in doing the update and in moving forward. Just to give you an example, we received comments by 1 individual, 17 countries, 2 intergovernmental organizations, and 29 other organizations. So combined, we received in all a total of 2005 individual comments for the peer review of this document, which we believe shows the great interest from various actors in the topic of synthetic biology. I would also like to note that through the process of the update, we were in constant communication, and we received also support from partner organizations like WHO, CITES, IUCN, because as you will know, when you go through the document, this latest version includes information about what some of those other organizations are doing in relation to synthetic biology, and therefore they were kind enough to work with the Secretariat and the authors in reviewing the text about their organizations and ensuring accuracy. When it comes to the scope and methods, then the scope, basically the authors attempted to create an apolitical document. As you know, technical series are technical documents, which is based on information from various sources. The other thing to note is that the authors use an inclusive approach. We know that there are divergent views about synthetic biology, many different opinions about the subject matter, what is not synthetic biology, but the approach that was used was an inclusive one. And then in line with this, I want to highlight that one of the biggest challenges that we faced through this process of drafting this document was actually related to the scope, and it is based on the fact that, as you know, there is not an agreed upon definition on what is synthetic biology. And because of that, that was also something that was pointed out through the peer review process. And we decided that we were going to use the operational definition that was produced by the ad hoc technical expert group on synthetic biology a few years ago as a starting point for this document. And that definition was, by the way, considered as a useful starting point for discussions by the Conference of the Parties as well. So we used that definition to frame, the content of the document. But at the same time, it's also important to highlight, and we want to underline that the authors are not championing any definition in this document. At the same time, also, the authors acknowledge in this document, and you will see that as well, that until consensus is achieved on what is or what is not synthetic biology, there will always be divergent views and opinions about this complex subject matter. Then when it comes to the method, we tried as much as possible to remain true to the request from the COP, which was to do an update. So as I explained before, we tried to identify areas, what part of the outline of the previous document could be maintained as much as possible and areas that could be kept. But at the same time, of course, we were conscious that the amount of information about synthetic biology has exponentially grown, in recent years. So of course, the outline somehow changed, but we tried as much as possible to remain true to that. So we removed text that was outdated, we added new text, and we updated some sections that just needed additional information. The main tool then that we use for doing this update, of course, was a bibliographic search, and we tried to keep that search as broad as possible. So one very useful thing was that we use as a starting point a bibliographic references list that was produced by the Secretariat also as an information document for SAPSTEP. And that list has been put together combining various publications and references that were suggested to the Secretariat through various processes over the intersessional period. So for example, when we had the online forum on synthetic biology, when we have the ATEC meeting, or submissions of information, parties and stakeholders submitted potential useful literature that we could use. So the Secretariat compiled that list and put it together on an information document. We also expanded that list with information that we have found ourselves. And that was a very useful starting point for us. So what happened is that we took that, and then over the final quarter of 2020 and the first quarter of 2021, we continued to do a bibliographic search, and we enlarged that list, and that was basically what we used for the first draft. And we went through peer-reviewed literature to great literature, like produced at all levels of government, academia, industry, civil society, and so on, in light of also, or linked to the approach that we said that was going to be inclusive for the document. Then important also to point out that once we had the peer review, of course, through the peer review process, many other useful references were suggested, and therefore that list continued to grow after the peer review, and the final one is the one that you can see on that publication. So I think that is a pretty brief overview of why we were doing the update of this document and how we proceed in doing it. I just want to say where you can find this document. Watson mentioned that it is now available on the CBD website. So if you go to cbd.int/ts, and that is for technical series, you will find the technical series 100 and all the other technical series that have been published by the Secretariat. At the same time, I want to let you know that there is a peer review page on the Biosafety Clearinghouse. The link is there. You can visit. You will find there the first draft, the final draft, the peer review comments, and the responses to those comments as well. To conclude my presentation, I just want to highlight the relevance then of these technical series number 100 for the CBD and its protocols. As you know, synthetic biology, it's cross-cutting topic, and therefore we believe that this publication is going to be useful not only for discussions under the convention, but also for discussions under the protocols. You might also recall that we have received a mandate from the Conference of the Parties to have a coordinated and cooperative approach when we discuss synthetic biology between the convention and the protocols, because precisely of this cross-cutting nature. So if you go through the document, you will see that there are areas of the technical series number 100 that directly are relevant for the Cartagena Protocol, Nagoya Kuala Lumpur Supplementary Protocol, as well as the Nagoya Protocol. Then the publication provides, we believe, useful information for the upcoming discussions. As you know, we will have synthetic biology discussions on COP15, so we believe that these will be useful for that, but not only for discussions under the convention, but also for discussions that could be taking place on other organizations and initiatives that are also discussing synthetic biology. And finally, we hope that this document then will contribute to the body of knowledge on this complex issue of synthetic biology. And with that, I will conclude my presentation, and thank you for your attention. So over to you, Watsi.
Thank you very much, Marianela, and thank you for providing a comprehensive overview of the process that we have undertaken and how this publication is helping not only the convention processes, but it's relevant also for the protocols. Next, we will now hear from Felix Moranta Barrios from ICGEB, and Felix will highlight the synthetic biology activities and growth since the Series 82 was published, and share with us the state of the art. Felix, your screen.
Yes, hello, Watsi. My screen, okay, should be there, isn't it?
Okay, we're waiting for your presentation.
Okay, thank you so much, Watsi. I will proceed to share my screen. That's it. Are you seeing my presentation?
Yes, perfect. Thank you.
Yes. Okay. Thank you, Watsi. Okay, good morning, everyone, and thank you again for your introduction. I am Felix, Senior Program Assistant in the ICGEB, and I'm going to cover the technical background on synthetic biology. And from now on, please keep in mind that, as Marianela told before, we have followed an inclusive approach. This could mean that the techniques, tools, applications, or products may not be considered as synthetic biology by all of you, considering the divergence of views. As we saw in the previous presentation, we have followed, and I insist on this, the AdTech operational definition of synthetic biology. This is that synthetic biology is a further development and new dimension of modern biotechnology that combines science, technology, and engineering to facilitate and accelerate the understanding, design, redesign, manufacture, and/or modification of genetic material, living organism, and biological system. And why is synthetic biology important? Well, look at this news from 2015. They were reporting a cutting-edge technique to edit mosquitoes' genome, for instance. It was the CRISPR-Cas technology. And now, look at this from the last year. We can see that the application of this particular technology is steadily increasing. And the same has happened with other technologies based on synthetic biology. Actually, while some research in synthetic biology is focused in promoting a greater understanding of the essential function of genome, for instance, most of the research is focused on commercial and industrial applications. And the global synthetic biology market was estimated to be valued at almost $7 billion and is projected to grow at this rate in the next 5 years. This can be attributed to the rising demands of products like synthetic nucleotides or oligonucleotides, as well as the increasing use of engineering technology for manipulating complex genomes, or the increasing development of therapeutics, and also the increasing technological advances in the CRISPR toolbox. But let's see now specific tools that are being used in synthetic biology. Synthetic biology relies in a suite of supporting technologies that enable the engineering and creation of biological components. This tool could include DNA synthesis, direct evolution, next generation sequencing, genome editing, engineered gene drives, RNAi-based tool like the RNAi interference, artificial intelligence, machine learning, modeling, also the use of biobricks, and recently the establishment of biofoundries. And synthetic biology also covers several areas of research, such as nucleic acid-based circuits, engineering of protein metabolism, and also of the entire genomes, protocells, xenobiology, or systems that are free of cells. Now I highlight in red the supporting technologies and areas of research that are new in this technical series. All the mentioned technologies, tools, and areas of research create a complex and rich scenario for developing new applications and products. We have divided them by its intended use. We have containment or contained industrial process or laboratory settings that refer to controlled and regulated environment. Also managed, semi-managed, or urban settings that refer to partially controlled or regulated environment. And we have the wild or unmanaged settings that refers to controlled or non-regulated wilderness environment. Regarding the degree of development, we have commercially available product that involves also final products that are distributed through non-profit enterprises and products approved for commercial releases, but which are not commercialized yet. Also near-term products that have already undergone a series of regulatory approvals to advance through the development, and research that also includes exploratory research and proof of concepts. Now, in the following slide, we are going to see some examples of this product. Let's focus, for instance, first in the contained the products that are produced in contained or industrial settings. In research, we have identified, for instance, this is just examples that I have extracted from the document. The development of minimal cells, non-native nucleotides and amino acids, the recreation of extinct viruses using chemically synthesized DNA, or for instance, the digital information storage using DNA molecules or biosensors. And near-term products, we have identified biofabricated wildlife products, cultured leather products, plant-based vaccines, or engineered probiotics for the delivery of medicine in vivo. And in this group of products, there are the most of the products that are commercially available, no? That it belong to biopharma products, fabric products, cosmetic and fragrances, and food ingredients that has been produced using synthetic biology techniques. In the next category, the managed, semi-managed, or urban setting, we have identified in the research level an intense effort to develop genetically engineered bacteria for the use in agriculture, the delivery of modified viruses using insects for the modification of crops, ongoing project for the extinction of extinct animals, and transient modification of agricultural plants through RNAi interference. Near term, we have detected genome editing crops and farm animals that are through regulatory processes, also engineered gene drive in mosquitoes and agricultural pests, and also self-limiting insects. And commercially available, we have two particular examples. One genome edited soybean and one biological nitrogen fertilizer that is based on engineered bacteria. Finally, for wild settings in research, we have identified, for instance, the research on genetically engineered bacteria for environmental application, such as bioremediation, biodegradation, or biomining, or for conservation purposes and control of vector-borne diseases, and for improving the resilience of wild animals and plant population. In comparison with the other two categories, here for the wild, we have not detected products that are near-term or commercially available. Now let's focus on what has changed in the technical series between 2015 and 2022. While other products in 2022, okay, most of the applications continue to be related to microbial metabolic engineering and are for contained use. That remains the same. However, there is a greater availability of high-value fine chemicals, which include nucleotide balancing or cannabinoids, for instance. This marks a shift from the biofuels previously profiled, and more industry have spurred new applications not covering in 2015. For example, food proteins, textiles, and materials that are being produced without the need of animal resources. Similarly, wildlife products such as rhinoceros horn could soon be replaced with biofabricated versions. Another change since 2015 is the development of commercially available products for environmental release, including the genome-edited soybean and engineered bacteria as fertilizer. And while other products are in advanced stage of development, such as self-limiting insects or genome-edited animal, for instance, others have progressed from early research, such as the LMOs containing engineered gene drive to control diseases. Further, in recent years, the application of artificial intelligence and the establishment, of course, of biofoundries have accelerated the research and development of synthetic biology. As you could imagine, the use of synthetic biology also triggers a wide variety of views related to risk, benefits, along with socioeconomic aspects and governance. And you will see a discussion on this during the following presentations. So many thanks for your attention.
Thank you very much, Felix. Thank you for that technical overview and for sharing with us all the exciting new developments in this field. And this is why I think we had such a large number of comments and interests, because the technology is advancing much faster than we had anticipated. So thank you for sharing that with us. We will now move on to the next presentation, which looks at the next set of chapters of the publication. And this will be made for us by Wendy Craig, our group leader and task master and task leader on this project. Wendy was also the project manager for this overall project, and we really appreciate your support, Wendy. But she'll be talking about the potential impacts from synthetic biology on conservation and sustainable use of biodiversity. So over to you, Wendy.
Thank you very much, Awazi, for introducing me. I'm just curious, before I start, that everybody can see my screen?
We can see your screen.
Good.
We can't see you, but we can see your screen.
Okay, maybe that's better for all involved. So anyway, I'm just going to dive in before I get caught up in any technical issues. I wanted to thank everyone for attending today's webinar, and also I wanted to thank the Secretariat for giving me the opportunity to talk with you all today. It was a great pleasure for ICGB to be involved in the construction and the drafting of this document, so I hope it meets everyone's expectations, although I understand that is a really big task. I'm going to spend the next few moments just going over what in the document is called Section C which, as Wadzi said, is the potential impacts of components, organisms, and products resulting from synthetic biology on the environment and the sustainable use of biodiversity. As we've just heard from Felix, there's a large number of techniques and technologies involved, and so it was not possible at all to cover every one of them in the granular degree that was necessary. So it quickly became obvious that only a small proportion of the available publications and information available actually analysed the extent to which potential impacts specifically from synthetic biology as a whole would avail themselves. So this really presented a challenge to us as authors, and so whenever possible, whenever we raised a topic in the document, we tried as much as possible to exemplify that with actual examples, though it really wasn't possible in many instances. So many sections of more generalized text have resulted, but we'd like to reinforce the fact that really potential impacts truly cannot be generalized in this way, and as such they should really be considered on a case-by-case basis, and they should really importantly be linked to specific uses. So as authors, what we tried to do was to prioritize those potential impacts that came from examples of applications that were either commercialized or in advanced research, or at the very least had received significant international attention, either through publications and literature or through discussions during international fora, such as the convention and other international meetings. So another caveat is that it's important to note that this section truly isn't meant to be an exhaustive list of potential impacts from every application of synthetic biology, but instead it focused on those that could be directly related to the conservation and sustainable use of biodiversity, which is the focus of part 4 of the document, and or to socioeconomic and cultural considerations, which was the focus of part 5, before it then turned to just general biosafety concerns, which was the focus of part 6. So as I understand, the majority of everyone in the webinar are really tracking my talk through the physical document in front of you. If not. Anyway, going into part 4, it was obvious that the operational definition of synthetic biology, which is described at the top of the document, really represents a wide array of potential impacts, and those range from benefits through neutral effects and ultimately to harms on biodiversity related issues. But in trying to describe such impacts, the authors felt that there was a multitude of factors that needed to be considered. One of the key things that we noted was that some of the potential impacts from synthetic biology are not unique to synthetic biology, and instead can also be applicable to any technological approach with the same aim. Such impacts will be specific to the host organism, while others may be related to the specific synthetic biology technique and the way it was used. Also, it was noted that some desired impacts from the technology are expected to have potential secondary effects on other species with, for example, common trophic bonds, pollination requirements, host pathogen relations, etc., which are totally independent of the deployed technology. And further, although an application may be beneficial in a certain social, political, economic, and/or ecological context, this should not really imply that it would also be beneficial in any other context. This therefore underlines the previous note that the potential impacts of each application should by necessity be considered on a case-by-case basis. So as we delve further into part 4, some of the potential impacts that we considered focused on, for example, this first one, specific applications that were being designed to modify, suppress or eradicate populations of various target, usually pests in this case, species, thereby increasing the feasibility of large-scale control, even with a potential for continental scale eradication, which is quite unique to some of these technologies, especially of unwanted wild populations or species. However, it's important to note that many of the examples discussed in the document remain hypothetical, as none have yet been widely deployed or released, and we'll come back to that later in the presentations. One area receiving intensive attention from developers using synthetic biology centres around the use of resources in agriculture, from strategies to reduce or replace chemical inputs to those that increase nitrogen fixation efficiency in crops and biotic bacteria, or that improve yields of current cropping areas through modified crop architecture. However, it's also been noted that some of these techniques may cause off-target effects, which may ultimately lead to unintended or unexpected consequences during interactions in the wider environment. And they would have to be evaluated during any risk assessment, which is a common element of decision-making when it comes to authorization of these technologies. The authors, we also noted that synthetic biology also has the potential to address some of the challenges resulting from climate change, especially with respect to coral reef restoration and the designing of next generation biofuels. However, neither of these are without wider environmental and market concerns, some of which also lends themselves, these concerns, to the replacement of natural materials, as also noted in the next slide. So, as we would imagine, the use of synthetic biology also triggers a wide variety of views related to perceptions of risks and benefits, moral and ethical values, along with broader issues such as socioeconomic aspects. And these were very much reflected in many of the comments that we received during the peer review process in the completion of the document. One element that completely struck us as authors was that society as a whole has a key role to play in helping decision makers and regulators better define specific scientific protection goals, i.e. that's the things that society doesn't want harmed, as well as to help decision makers and regulators explicitly determine the extent to which any harm may be acceptable or not before they consider giving any authorization of the use of these technologies. Further, it was recognized that soliciting Indigenous peoples' perspectives and also traditional perspectives on synthetic biology and being able to coherently integrate them into decision making is a persistent challenge. But there has been some movements on this front that others can be able to learn from. Further, different fundamental objectives of international trade and environmental regimes have led to differences in regulatory measures. What we found as authors that there was the need to strengthen the coherence of these two systems, and that will require measures to be taken at national and supranational levels to ensure that they are implemented in a mutually supportive manner. And once again, society will have a key role to play in this debates and activities. Further, ethicists have been actively engaged with the new tools and techniques of synthetic biology for more than 20 years now, and the ethical debate on synthetic biology is further exacerbated by the novel mode of action and the increased accessibility and the speed of use and uptake associated with the technologies and platforms that Felix described. Therefore, common considerations have included the ethical debate on whether to ban publications of dual-use science discoveries, and extended to whether synthetic biologists are even playing God. Much of these elements are explored further in the document, but for brevity, I'll move on to the next part, which concerned general biosafety concerns. And these relate to any accidental or intentional release of organisms resulting from synthetic biology. These concerns and the way that they may be addressed under biosafety systems will vary and depend on the type of organism and trait, the intended use, and the receiving environment. Thus, the adequacy of current methodologies for environmental risk assessment of synthetic biology products will very much depend on the degree of novelty and complexity exhibited by these organisms and applications. Some of these applications are already challenging existing regulatory systems due to the need to address these novel risks and impacts, as well as the levels of uncertainty and currently untested mechanisms for observation and monitoring. Together, these are compounded by the ever-increasing pace of development of these technologies, as Felix highlighted. Regardless, many regulatory authorities are beginning to present their various approaches to determining the risk presented by specific synthetic biology applications, especially with respect to those using engineered gene drives, genome editing, and risk-based technologies. Thus, should in an environmental risk assessment identify any potential impacts on biodiversity associated with these applications, then it's obvious that strategies to mitigate and manage these risks should be developed. And these have so far focused on removing unwanted inserted sequences, the use of virulence factors and synthetic resistance. Others have looked at more genetic containment approaches, which in the past have proven to be quite contentious, as well as the possibility of post-release removal of the organism from the biodiversity, as well as them being tractable or traceable in the environment through various detection and identification mechanisms. So in a nutshell, I've tried to highlight how the use of this synthetic biology can trigger a wide variety of views and that's related to risks and benefits, along with socioeconomic aspects and governance. And hopefully I've given you just a taste of what you can find and explore on your own cognizance further in the document. And with that, I would like to now pass over to Rodrigo to present on the next section of the document. Thank you.
Thanks, Wendy. Hello, everyone. Just a moment. I'm just waiting for a function to appear that allows me to share my screen. Okay, which should now be available. And just apologies, but I'm working from a new workstation, so double checking that you're seeing a PowerPoint presentation and not my other screen.
Yes, Rodrigo, we can see your presentation.
Excellent, excellent. Okay, we're good to go. And I'm mindful, I think we're just a little bit out of sync with the initial timetable, so bear with me. I'll do my best to get us through and stick to time. So, hello everybody, I'm Rodrigo Sara. I'm a commercial and IP lawyer. I specialize in responsible bioinnovation and technology transfer. I thank the Secretariat and also ICGEB for the opportunity to join today's presentation to socialize the governance and regulatory perspectives that are covered in the updated technical series It's been a fascinating process to be involved in the update. As was mentioned, it was over a year of work in the pipeline, responding to thousands of comments. And as authors, I don't think when we started, I don't think we perhaps appreciated just how much had happened in such a short period of time, that the update would require such comprehensive, would be much more comprehensive than we had initially envisioned. So today's focus is really just to give you a tour to socialize Section D in particular, and on governance and regulatory perspectives, starting just with an explanation of the rationale. We had the fortunate forensic analysis in the initial technical series that we've built on. So we've just built on the existing comprehensive coverage of synthetic biology in the context of the conventions and its protocols. We have then also undertaken a comprehensive but yet not exhaustive review of how other international organizations and initiatives are discussing synthetic biology. And we deep dived into 3 particular initiatives that have a substantial program of work on synthetic biology. So that's the rationale in terms of the approach for this particular section. And just to highlight some differences with the previous technical series. So as mentioned, we have updates on the convention and its protocol, extended coverage, and this section, the extended coverage has undergone significant revision because so much is happening in this space, and we felt it would be useful to really deep dive into a couple of initiatives that wasn't the case in the original technical series. So you hopefully have had a chance to look at the outline. It commences with it sets the scene by looking at the governance and regulation of synthetic biology. It builds on coverage in the technical series, including self-regulation by the scientific community and moratoria, and new initiatives that are underway in that space, as well as updated considerations on IP issues related to synthetic biology and biodiversity, including proprietary and non-proprietary approaches. Then it builds on this forensic analysis of and coverage of the potential implications of the convention and its protocols, looking at specific provisions and what they may mean from the perspective of synthetic biology. So we've just built on that work to update it, but it hasn't undergone extensive revision beyond highlighting the latest decisions in COP13 and COP14 that weren't covered by the previous series, looking at the work of the relevant RTEGs and under the different of the various RTEGs that are in place, looking at substa recommendations, looking at SBI, and all the way leading up to COP15. And then what I will do today is give you more of a tour, socialize these elements that have undergone more significant revision, which is focusing beyond the Convention on Biological Diversity and its protocols, and really looking at the governance and regulatory landscape under other treaties and initiatives. And here we, building on we can see that the decisions in COP13 and COP14 are quite substantive, and so that even within the CBD, the program of work has amplified, and there are a lot of activities underway, and all leading up to COP15. But then under other international instruments and mechanisms, we've provided, in some cases, updated coverage, and in other cases, more new coverage or deeper coverage. And here I'm just flagging with the stars elements that have undergone substantial revision. So I mentioned deep diving into 3 particular initiatives. We'll hear more on those in a moment. We've also updated, provided more of an update on what's going on in the Biological Weapons Convention, under the Environmental Modification Convention. We've looked with a fresh lens at the UN Declaration on the Rights of Indigenous People and related initiatives, and also at what's happening under the instrument that's being negotiated under the law of the sea on the conservation and sustainable use of marine biological diversity outside of areas of national jurisdiction. So that's just to give you a bird's eye view of elements that have undergone significant change. And focusing just to provide and socialize a little bit of just an overview of these three particular initiatives that we've provided a deeper dive on. So we have seen that the World Health Organization has quite a number of initiatives underway that are relevant from the perspective of governance and regulation of synthetic biology. They are undertaking a review of the health of their international health regulations following the COVID-19 pandemic, but that is specifically looking at issues that are relevant to synthetic biology. They also, in 2016, published updates to recommendations concerning the distribution, handling, and synthesis of the variola virus. And that was following a process of extensive consultations on issues of biosecurity and biosafety related to synthetic biology. They've also updated their laboratory safety manual, and which now includes a section dedicated to synthetic biology as one area of emerging technologies that need to be considered. So there are even more interesting. There are other initiatives underway by the WHO, and they're covered in more deeply in the technical series. It's just to give you a flavor. Likewise, many of you may be familiar that the International Union for Conservation of Nature has also expanded its program of work and is related to synthetic biology. And so that commenced in 2016 with a resolution calling for an assessment of issues related to synthetic biology that may have an impact, whether positive or negative, on conservation and sustainable use of biological diversity. And that call was for an evidence-based assessment. And there has been a significant amount of work through the different bodies of the IUCN on that resolution, culminating in 2020 on actually a motion which has been approved to establish more of a concrete roadmap towards an IUCN policy on synthetic biology, which is quite significant in terms of the landscape outside, what's happening outside of the CBD. Likewise, the Convention on International Trade in Endangered Species of Wild Fauna and Flora, so CITES, in 2016, it also commenced the process to review its provisions and resolutions as they might relate to specimens produced from synthetic or cultured DNA. And this has been quite an extensive process involving commission studies and interestingly culminating in a call to strengthen coordination with the Secretariat of the Convention on Biological Diversity, also with FAO, and also for the International Union for Conservation of Nature and other relevant organizations, which will feed back into some of the key messages that we have. So hopefully that has given you a socialized view with the content. It's quite as I'm sure you've seen, it's a comprehensive document. There's no time to dive deeper into the granular coverage, but hopefully this has given you a good perspective on regulatory and governance issues related beyond in the CBD and beyond. Thank you.
Thank you very much, Rodrigo, for highlighting some of the new elements that this series picked up on, and in particular, how the package of interventions all comes together. And I think through this process, we were also able to strengthen our own relationship with the other international agencies. So it was a very worthwhile exercise. The next presentation is a key presentation, and I think it has the take-home messages that we would like you to have from the technical series. And so I will hand back to Wendy and Rodrigo to tag team on the key messages and conclusions from this technical series. So over to you, Wendy and Rodrigo.
Okay, hopefully now I have overcome my technical incapabilities and that everybody can not only see me, but also my screen. Anyway, and okay, well, I don't know that it's perfect that you see me, but anyway, I just wanted to thank you once again for giving me this opportunity to come back on screen to go over some of the key messages that we felt as authors that much of our exploration of the various literatures and international discussions had led us to. For the purposes of timing, I won't go into every one of them, so probably the audience is very grateful for that, but I just wanted to highlight a couple of them. So what we try to do as an authorship, we try to group the various messages into 4 main themes, and we present them here on the screen. So the first 5 messages focus on the current state of synthetic biology. Before then, a couple of messages focus on the potential impacts from synthetic biology as we saw them. Before then, another 2 or 3 messages then looked into more depth into communication and engagement and transparency before turning to the last messages covering synthetic biology, regulation and governance. The first 3 points I'm going to cover, and then I will pass back to Rodrigo to cover the last ones, as well as some other gaps and overlaps and conclusions that we identified as authors. So the first message, which you can see here, is that we recognize that synthetic biology is a cross-cutting and rapidly advancing discipline that's having increasing relevance to the environment, to food and health, and amongst many other global challenges, which some of us have raised in previous presentations here. We also heard from Felix that there's such an expansion of the diversity of innovative tools. The number of products and potential applications of synthetic biology is ever increasing, and it's leading to advances in industrial products and to improvements of plant and animal engineering, personalized medicine, and many other different sectors as well. And so it's really offering so many great opportunities for us to address current global challenges to the extent that it could help contribute to achieving a multitude of different United Nations Sustainable Development Goals. So that's one of the positive things that as an authorship that we recognized. Also, moving on to key message number 2, it was recognized that that there's still no consensus on which techniques or applications will ultimately fall under the definition of synthetic biology. And the consequence of that really is that there will remain an array of divergent views in this respect that will ultimately pose challenges at various levels that range from the governance of the synthetic biology to more specific aspects such as problems in applying requirements regarding current or future regulation, amongst others. But what we felt it should be worth noted was that even in the absence of an agreed definition, there is still an interest and a will at various levels, albeit national, regional, or even international, to take advantage of these new technologies, including synthetic biology, in a responsible manner, and that could ultimately maximize potential benefits while also minimizing the risks. So then there was also message number 3, that was that with early applications reaching such large-scale deployment, that we've seen the economic value of the synthetic biology market continues to increase. And I'm not going to go further into that message. Message 4 was that despite its potential global deployment, research and development in synthetic biology mostly occurs in only a limited number of countries so far, and so only a limited number of countries are gaining the benefit from these applications, and it could be counter-argued that any potential negative consequences of these technologies and applications are still limited at a global context. Should there be the desire, the interest to increase the number of countries that where this research and development is occurring, then this will require initiatives to be designed to facilitate technology transfer, to improve scientific cooperation, and to help capacity building. Also, it will require cross-border challenges and collaboration to be overcome, and to improve access to cutting-edge technologies in such a way that they're leveraged to enhance the research, the development and the deployment in such countries that currently only have a limited capacity in synthetic biology. Furthermore, we saw that the constraints currently from the intellectual property domain will have to be further explored, and a fostering of open science to help support scientific cooperation and to make science a more transparent, accessible, equitable and inclusive. It would be really useful, and it may assist in narrowing the technology divide observed in relation to synthetic biology. Lastly, on this slide, we recognise that while most applications in synthetic biology so far have been within contained industrial or laboratory settings, this is changing rapidly with an increasing focus on managed or semi-managed or urban settings, and in the longer term, a potential for use in unmanaged and wild settings. So now we're going to look at the group of 2 messages that looked at the potential impacts from synthetic biology. The first one of which was message number 6, and here we identified that the potential of the synthetic biology toolbox is vast, and as we heard also from Felix, it's ever evolving also, and so is the potential for impact on various sectors. It's worth noting, however, that even though there are numerous synthetic biology applications under research and development, and many that have reached the market, only a few applications developed for direct use in the environment have been authorized to date. This means that there is still relatively little field data collected concerning their potential impacts. Therefore, except for a few specific cases where releases have been authorized, the range of potential impacts of these applications on the conservation and sustainable use of biodiversity, it remains largely based on theoretical analysis to date. Thus, such discussions on potential impacts have only been informed by a limited number of small-scale experiments, mostly in the laboratory, and by previous experience with, for instance, LMOs with similar traits. So this obviously brings challenges for achieving consensus on whether and how synthetic biology applications are to be assessed and regulated, and whether any regulation should make use of the same regimes as those in place for LMOs, with or without adaptations, or whether new regimes should be developed. The other message that we identified in this grouping was message number 7, and we noted that many of the assessments of potential impacts that were originally anticipated, maybe even described in the previous version of the technical series back in 2015, were overly simplistic in nature, while more recent information has revealed greater complexities, suggesting that there's a need for a more nuanced approach in exploring the extent to which these impacts could potentially result. And finally, at least from my side, I like to focus on those messages that we grouped under communication, engagement, and transparency. And the first one of these is message number 8, where we noted that early society and community engagement in synthetic biology research is a key component of transparency. So here we recognise that the global nature of synthetic biology applications and the fact that local communities are likely to be the first that are impacted. It would be advantageous to communicate the concept of new applications to those communities prior to any large investments of time and resources. We also believe that early engagement with potentially impacted communities, including the IPLCs, could provide opportunities for discussions related to potential benefits, risks, and concerns, and that sufficient broad and inclusive discussions could in turn improve public trust and policies that reflect a broad range of stakeholder views, as well as assisting with the full and effective participation of IPLCs. And then I'm just going to turn to the final 2 for me, and that's messages number 9 and 10. So number 9 focused on the greater public engagement and regulatory decision making is needed in order to ensure that regulatory practice best meets societal expectations or desires or goals. And lastly, for me, that biosecurity risks arising from synthetic biology could be mitigated through improved transparency, communication, and self-regulation. And it's at this point now I will pass over to Rodrigo, if possible.
Thanks, Wendy. I'm sharing my screen, but unable to see whether it's appearing on your end. Just a confirmation, please. Yes,
we
okay.
can see it, yes.
Yes, excellent. Okay, so just down to the last 3 key messages, all of these addressing or in the context of regulation and governance, and there's some overlap between them. So, I mean, one of the key messages that emerges is this is an increasingly complex space when we look at the governance and regulatory implications for synthetic biology. And so a key message is really there is a need for an enabling policy and regulatory environment. There's a possibility that the current complexity is not conducive to an enabling policy and regulatory and government. So that's one key message. Another is that because of this complexity, there's really a need for more coordination and cooperation across the different fora where governance and regulations are occurring. And to address this complexity, to minimize this complexity, and to create a more enabling policy and regulatory environment. And then finally, it's really horizon scanning and foresight. There's a real need to look ahead, to anticipate what kind of developments will occur and what will be needed from a regulatory system perspective to future-proof the fast-evolving regulatory environment, to make sure that it's able to keep up with the fast pace of the technological developments occurring in synthetic biology. So that's it for the key messages. And now we'll just provide an overview of the synthesis of the technical series. It provides a coverage of observations, analysis, and conclusions. And sorry, I hope I haven't just stopped sharing my screen. No, okay.
So we have it. You've just stopped the presentation format.
Okay. Oh. Okay. Apologies. I'm just trying to. Well, actually, just bear with me, so I don't take any more time. So, just to give you a high-level coverage and some insights. So, firstly, as has been acknowledged by Wendy and Felix, this is a multidisciplinary area of research, and that disciplinary intersection and then the underlying complexity creates really significant challenges for effective governance. So that's one key observation. the other is acknowledging when we look at this proliferating regulatory landscape, that there is a real risk of gaps and overlaps arising across these international instruments and regulatory mechanisms, and in a number of areas. So whether it's risk related to harm to health, environment, biodiversity, and human welfare, whether it's around conservation and sustainable use of biodiversity, whether it's concerning access and benefit sharing, including how to strengthening capacity development and technology transfer related to synthetic biology applications, and also commerce and trade. So we see, I mean, these are just some thematic areas where we see a significant potential for gaps and overlap to arise because of the complexity in this regulatory landscape. And so this really calls for a more, really a need for a more holistic approach for international governance. There's a real risk that with the extent of potential gaps and overlaps that international frameworks are operating or in silos, or there is a real risk if they do operate in silos in the absence of coordination. And it's a real challenge, how to overcome these silos to ensure strengthened coordination. And one of the observations is that the existing mechanisms, given how long the Convention on Biological Diversity and its protocols have been looking at this issue of synthetic biology, it provides an opportunity to leverage improved coordination and to strengthen coordination with other international fora. Another observation is that there's a need for regulation and governance that strikes really a delicate balance between the risks and opportunities presented by synthetic biology. So whereas on the one hand, of course, there's a real imperative to mitigate risk, to health, to the environment, to biodiversity and human health, human welfare more generally, but there's also a need to realize where that risk can be adequately mitigated to ensure that the potential benefits of synthetic biology, for example, as they might contribute to the UN Sustainable Development Goals, are realized. So there's this delicate balance between risk and opportunity that needs to be addressed. And then finally, I think it comes as no surprise that the key observation and our personal our reflection as authors is that it's the technology is is evolving, even from even now it is out of date just because of how quickly. I mean, the aspects of this technical series have been overtaken by developments in synthetic biology research and deployment. But likewise, the regulatory environment is also evolving very quickly. The review of the regulatory landscape is as I mentioned, comprehensive but not exhaustive. It's if we were to look at this 2 years ago, we'd have a very different content for the technical series. If we were to look at this 2 years from now, we'd again have very different content, just because of how quickly both the technology and the regulatory environment is evolving. And and just to wrap up this work, it's not just of the authors, but also the broader regulatory science group at ICGED. And so a big thank you to them as well for their support. And that's it from me.
Thank you very much, Rodrigo and Wendy, for those key messages. And they are available on the website if you would like to look at them again further. We are running a little behind in terms of time, but we do want to hear some responses from a number of stakeholders and interest groups on this very important topic. So they will share some perspective with us very briefly, and just 2 minutes each, just to hear and get a sense of how this document relates to different stakeholder groups and how they see it helping them in their own different perspectives. So we will start with Anita Anthosamy, who's the Assistant Director at the Department of Biosafety in the Ministry of Environment and Water in Malaysia. So Anita, over to you for your very brief response to this technical series. Thank you, Anita.
Thank you. My name is Anita, so I'll skip the introduction. So my first comment on this technical series is the availability of the document per se. So this document, as you heard from the first presenter, is a result of consultation that was not limited by borders and utilized the Secretariat's wide reach 2005 individual comments. Wow. So now if we were to initiate and produce such a comprehensive resource like this ourselves, there are limitations of resources, availability of experience, expertise, and even time. So now through this document, we have leaped into an advanced starting point to identify and address these issues related to synthetic biology with all the baseline information in our hands. This document, in my opinion, is versatile and can be used for many different purposes, whether to initiate a consultation, coordination of enforcement mechanisms, public consultation, possible consideration for risk assessment, or even socioeconomic consideration. So the availability of this document as a reference point and a peer-reviewed resource material will help to identify and tackle the issues in whatever means needed. There is a need to be prepared for a possibility of addressing commercially available SynBio products with requests to be placed in the local market. Now, even if a country is not a producer, there is a high possibility that we may be a recipient or even importer of SynBio products which will eventually reach our shores. Our current regulatory mechanism doesn't specifically address all issues related to SynBio, and this results in gray areas in regulatory scope. There is a need to have conversations with many different stakeholders to future-proof the existing regulatory mechanism and approaches. So a good reference point for stakeholder consultations, I mentioned, would be the technical background, which is section B that was covered by Felix. This section provides an elaborate description of synthetic biology. So based on our experience in doing consultation, we find it essential to provide a good explanation of the technology, to provide an accurate context to stakeholders, to initiate a more productive discussion, and ensure that everybody is on the same page in their understanding of the technology. Nowadays, you can find information anywhere, Facebook, TikTok, whatever. However, the information might not necessarily be factually accurate, and this may influence the progress of discussion if stakeholders do not have a good understanding of the technology. So back to this document. This section provides easy and simple to understand language and carefully avoids jargons in the explanation. So a good supplement to section B is table 1 that provides a good and quick overview of synthetic biology, broad categories, and settings of use or intended use. So this information further splits into what is the current phase. So this gives us an idea of how soon this is going to become a reality. So theoretical information is translated into reality, and this helps us to see the relevance of how SynBio can have an effect with clear examples provided. So why do I think this part of this document is important? Okay, so this will answer the questions that we usually hear when we call for consultation. How is this relevant to me? Sometimes it's hard to see the connection. So this part of the document gives a clear picture of how it may be relevant and why this issue of synthetic biology involves a diverse range of stakeholders. So speaking of the diverse range of stakeholders, this brings me to my very last point. Part D of this document is useful to identify the cross-cutting regulatory scope and issues that may be addressed by different policies, legislations, and regulatory agencies. It provides examples and options of various regulatory approaches practiced. So the consolidation of international laws, processes, and initiatives addressing synthetic biology, specifically in parts 9.2, 9.3, helps in identification of stakeholders who are relevant to discuss this issue. So this information is further supplemented. I refer to table 3 that provides a great overview of the information. Time permits me to highlight and elaborate only a couple of key elements from this document today. I encourage you to take a look for yourself and see everything this document has to offer. Thank you for listening, and thank you to the Secretariat for the opportunity.
Thank you very much, Anita, and thank you for highlighting the different parts of the document that are useful from your perspective. Next, we will hear from Lillian Chimpepo, the Principal Environmental Officer and the Biosafety Focal Point for Malawi, and we will hear from the protocol perspective. Lillian, over to you.
Thank you very much. Let me thank Secretariat for this timely development of the technical series on synthetic biology, especially now that discussions are still underway on synthetic biology at CBD level. We are mindful as developing country of the limited capacity that we have in synthetic biology, and therefore we look at this technical series as a much needed document that will bring us the much needed capacity that we need for the subject matter. And this will surely improve our understanding on synthetic biology, which will enhance our effective participation during the ongoing negotiation on synthetic biology. Secondly, I want to comment on DSI. DSI is also one of the technical, subjects that is under discussion and very important at CBD. And when we look at synthetic biology, it also uses DSI. So while parties are negotiating the modality to ensure fair and equitable sharing of benefits arising from the use of DSI under the Nagoya Protocol and the CBD, the Cartagena Protocol is much interested in ensuring use of DSI in synthetic biology should not cause adverse effect on biodiversity and also risk on human health. On the aspect of governance, which is also very key as regulators, we acknowledge the importance of synthetic biology as one of the biotechnology which has potential to offer benefits in addressing global challenges in agriculture, health, and climate change. However, as all technologies, they might have potential adverse effects, which were also presented during this launch. It might also even be subject to misuse, which can cause biosecurity and also health concern, public health concern. And this therefore calls for the need to have a robust biosafety legal framework for effective regulation of synthetic biology and also their product. The scientific knowledge which has been provided in this technical series, including the key messages that have been highlighted, will provide the necessary basic knowledge to guide the development of biosafety legal framework for those parties which don't have, and also for those who are updating their biosafety legal framework, like the case of Malawi. We have a very old biosafety legal framework which did not consider synthetic biology by that time because synthetic biology was not widely used then. And therefore, we consider this technical series as very important as we review our biosafety legal framework. We are also mindful also that the technical series will also help us as developing countries with limited experience to better understand the broad and regular horizon scanning process for the new emerging technologies, including synthetic biology, which is being established under the Cartagena Protocol on biosafety. The content of the technical series is not only relevant to us as regulators, but also you look at the scientists, researchers, including the academia and the private sector, and even the general public, who are the end users of the products that will be generated through synthetic biology. And therefore, it provides the scientific information behind synthetic biology, and also gives the in-depth information on synthetic biology tools which are being used in synthetic biology, and also the cross-cutting nature, including the benefits and the potential threats. This indicates that the approach which was used in developing this technical series was unbiased and therefore highly recommendable. And therefore, the technical series on synthetic biology qualifies to be as one of the key reference material on this subject matter under the convention, and also it's all the protocol, the Cartagena Protocol on Biosafety, as well as the Nagoya Protocol on Biosafety, and even beyond. Thank you very much.
Thank you very much, Lillian, and thank you for highlighting the relevance to other international discussions at the moment. The next speaker will be Kamal Rai, and Kamal is a researcher in indigenous science and philosophy from Nepal, and he will share with us an IPLC perspective. Kamal, over to you. We can't hear you, Kamal.
Thank you very much. I would like to thank the Secretariat giving me this opportunity to say something from the IPLC perspective. So for the IPLC perspective, this document, I think is a good structure, and the IPLC, their concern, their human rights, their participation, the knowledge system is in the document. So for this, I think it is a good document to discuss among the IPLC for the capacity building, for the awareness building. Also, it's very important to discuss this the GBS the post-2020 global biodiversity framework discussion. So for the IPLC, it's very important, the synthetic biology and its related technology. So we are, since beginning, we are more concerned about its development and its further progress. To understand, because of this, the technology and the science is very fast moving. For us, it's very difficult to follow their process. And so in these regards, for IPLC, their full and effective participation is very important. The women and youth from IPLC need to focus for the partner participation is the development and its research or any other progress. For us, the nature is alive and sacred, and it is the mothers. We are more concerned about the adverse effect of the synthetic biology and its other development. So for this reason, we need to more the strong regulation and precaution approach. So I found some of these precautionary or regulatory approach in this document. So we can carry and we can discuss. We use this as a technical series for our purpose. For more about this, the synthetic biology and its for other related the development is very important for us to understand its impact. For example, the gene-drive mosquito. Well, we know they are in the lab, but what is their behavior? What is their negative impact when they release in the wild within their wild species, and their behavior, the attitude to other species? They may bring some others unwanted impact on the nature or an ecosystem. That's impact for our water ecosystem or our food ecosystem that the indigenous peoples and local community, women and elders rely on that. So we need to know in detail, and we need to be engaged, and we need to collaborate. We need to understand the science and technology of synthetic biology and its related development. I thank you. Thank you very much.
Thank you very much, Kamal, for sharing that very important perspective with us. And we did make some attempts in this review to try and capture some of those ideas, and we're pleased that you found it helpful. Our last respondent will be sharing a civil society perspective, and we will ask Lim Lee Ching from Third World Network to provide us the final perspective. Lim, over to you.
Thank you very much, Wadzi, and thank you for the opportunity to provide some reflections on the updated document. I think the first point I want to make is that governance based on the precautionary approach that takes into account the rights of indigenous peoples and local communities is essential for ensuring that development of the field proceeds in a sustainable manner and is supportive of environmental and social considerations. Secondly, we reaffirm the document's assertion on the need to expand the focus of governance beyond biosafety, human health, and the biophysical environment to a more holistic approach that also encompasses social impacts, ethical principles, and elements of social justice. These are necessary elements for technology assessments, and efforts are needed to further provide the tools for parties to be able to adequately assess these technologies and their impacts. The third point I want to make is that the assertion of increasing economic value of synthetic biology markets is not as relevant as providing the basis for anticipating the potential adverse effects of synthetic biology. In order to do this, technology horizon scanning, monitoring, and assessment is critical, and the elaboration in the document of the applications in near-term research and commercialization and their potential impacts are a useful foundation for such a process. Fourthly, the assessment of potential impacts is an obligation under the CBD, so it's not a question of whether to assess. And while it is correct that only a few applications developed for direct use in the environment have been authorized to date with low field data available, this does not mean that the impacts cannot be assessed. Experiences with risk assessment for LMOs remain relevant, and the novel and sophisticated techniques that are used to produce the synthetic biology organisms can also be used to risk assess those organisms. Fifth, the document rightly points to the need for greater societal and community engagement and participation, including the need to obtain the free, prior, and informed consent of IPLCs. So as parties to the CBD, there is obligation to put much more effort in this regard to align with the many decisions and guidelines that the CBD has already adopted on these issues, as well as the guard against real and apparent conflicts of interest. Sixth, regulation and not self-regulation is essential, particularly given the dual-use nature. Parties to the CBD and the Cartagena Protocol already have legally binding obligations to regulate LMOs resulting from biotechnology and modern biotechnology respectively. So we believe that the CBD and its protocols are currently the best home for the international governance of synthetic biology. Of course, this is complemented by other international agreements that have mandates on specific aspects of synthetic biology and whose governance thereof should align with CBD principles. Finally, as this is a living document, of course there remain opportunities for improvement. We would like to see, for example, future sections dealing with cell-spreading GM vaccines, which are developed for intentional release into the environment and have a high degree of complexity and uncertainty, which warrant further scrutiny. Additionally, it's important that there is continuously updated knowledge. For example, with regard to genome editing, research demonstrates unintended on-target effects that have implications for safety and efficacy. So we think that this document is useful and should be continuously updated in order to keep up to date with the science and to ensure that synthetic biology applications are robustly assessed and regulated. Thank you very much.
Thank you very much, Lim, and thank you for even demonstrating that there is still ongoing work that we need to do in this regard. And at this point, we are running a little bit late, 9 minutes late to be precise. So before we look at the questions, we've been joined by the Deputy Executive Secretary for the convention, and he will now launch this publication on our behalf. As you know, the goal of the CBD Technical Series is really to contribute to the dissemination of up-to-date and accurate scientific information. And when we have a technical series like this updated, it's only right that we have the requisite fanfare. And to celebrate this, the Deputy Executive Secretary will now make his launching remarks. Thank you, David.
Thank you very much, Wadzi, and yeah, I'm really honoured that I have this job. I'm really quite excited about this publication. I'd like to thank you, but also Marinella, Felix, Wendy, and Rodrigo now for some really excellent presentations. Also to Anita, Lillian, Kamal, and Li Qing for some really very thoughtful comments. And also to everyone actually listening to this webinar for your attention and your interest in this event. I hope it's been useful in understanding what this technical series number 100 on synthetic biology can do. Certainly the task that the authors had in developing this updated version was not easy, and I think that's probably been clear from today's presentations. We can see a lot of the challenges that they were faced when it comes to the scope of the document, ensuring an update of an earlier version while ensuring that the latest information is available. There were many long hours of research, drafting, checking, back and forth, and I think though that this effort has paid off. I want to thank all of those involved in preparing this, the IGCEB and all my colleagues in the Secretariat in particular, but also all of those that were involved in reviewing the document. We had more than 2,000 individual comments from various actors, and I think that shows the interest in this from our parties, from the stakeholders. It's a topic, I think, of interest, a very broad interest across developed and developing countries, but also in industry, across indigenous peoples organizations, local communities, and other civil society organizations. It's clear that this topic of synthetic biology is a really important one for the Convention on Biological Diversity, and also for the Cartagena Protocol on Biosafety and the Nagoya Protocol on Access and Benefit Sharing. Some of you have mentioned just now its importance also as we come to the final stages of developing the post-2020 Global Biodiversity Framework, which will be adopted at COP15. And of course, COP15 will also address explicitly the issue of synthetic biology, in particular trying to respond to the call from COP14 to develop a broad and regular approach to horizon scanning. And I think this publication should be really useful in that. Hopefully it will help us remove some of the brackets from the decision, the draft decision that will be before the Conference of the Parties, but certainly also hopefully be a good starting point, a good resource for that broad and regular horizon scanning, assuming that the COP agrees for that to go ahead. So many of you have also referred to the fact that this is an area of fast development of new technologies. and that we need to be able to continue to update. This needs to be a living document, and that also we need to be able to continue to react not only from a scientific point of view, but from a regulatory point of view in a way that engages all the stakeholders. I think this publication, as many of you said, provides a good starting point in all of those areas. And again, I really want to thank all those involved. I'm really proud of the team that developed this. I'm honored now to launch Technical Series 100 on synthetic biology. Congratulations and thanks to all of you. Back to you, Wadzi.
Thank you very much, David, for officially launching Technical Series 100. It's available on the CBD website, and this webinar is available on the Biosafety Clearinghouse if anybody wants to listen to the presentations again and get the key messages. Unfortunately, we ran out of time and couldn't respond to all of the questions during the webinar, but we will be responding to the questions via email and making the questions and the responses available also on the webinar recording. So thank you very much, everyone, for joining us, and thank you for all of the effort that has gone into putting this together. It's truly a CBD family publication with the comments that came from so many of you, not only demonstrating the strategic importance of the topic, but also the process in itself, how we have all benefited and developed an even bigger repository of references from a number of sources. So thank you to everybody, and let's hope that this technical series will advance the discussions and support the processes underway. So thank you to everyone, and goodbye.